Welcome to the online presentation of poster 553, the benchmarking of 4C-seq algorithm combinations unpublished in simulated data. I will first give you a short introduction into the topic, and I will do this by starting with the role of chromatin conformation changes in cancer. Chromatin organization plays a crucial role in gene regulation, and dysregulations can be associated with cancer development and progression. The good news are that chromatin modifications are potentially reversible and therefore are potential targets for novel cancer therapeutics. Chromosome conformation capture sequencing, short for 4C-seq, is a cutting-edge next-generation sequencing technique which reveals the 3D structure of selected genomic loci and helps to identify chromatin loops between genes and regulatory elements. The analysis of those data is highly complex and prone to biases, and right now there are several 4C-seq algorithms available, but benchmarking revealed that none of them perform sufficient in all main use cases. Therefore, our aim was to exploit potential synergies between existing algorithms for optimized analysis. To reach this aim, we developed the following workflow, which you can see here.
The workflow can be divided into 4 steps: the pre-processing, the interaction calling and formatting, the centerpiece of this workflow, the ensemble algorithm, and then the performance evaluation. In the pre-processing, the FASTQ files, in this case from public and simulated datasets, are processed so that they have the correct input format for the 4C-seq analysis tools, which are then executed in the next step. The interaction calling and the formatting. The tools we tested were 4CeQ, 4seq, 4C-seq, R3C-seq, and PIXI, but unfortunately the output format of those tools are quite heterogeneous, and this is why we have to bring them into a standardized format. Then the results of the individual tools, which are now in a standardized way, are combined in different ways in the ensemble algorithm step. In this step, I used intersection, union, and majority vote to combine the results of the individual tools. Furthermore, I want to determine what are the ideal weights of the individual tools for a great performance in interaction calling. For this purpose, I used Bayes optimization based on the F1 score. In the final step, the performance evaluation, I evaluated the performance of the individual tools and the ensemble strategies using validated interactions and metrics such as precision, recall, and F1 score. The performance evaluation leads to following results, which I will show you now in the results section. I first focused on the performance of the individual tools and compared them regarding precision, recall, and F1 score, which I plotted on the right. And what we can see is that R3C-seq had the highest median precision and F1 scores, while the cis interactions of 4C-seq had the highest median recall. The other tools had a much worse F1 score, and now I will show you the performance evaluation of the tool combinations. And here I only show you a selection of combinations I tested because I tested a lot of them. And yeah, we can see that most of the tool combinations are able to outperform the individual tools except for R3C-seq regarding the F1 score. And it seems like the inclusion of R3C-seq into a combination seemed to work as kind of precision booster. And therefore, even an intersection, so even a simple intersection of R3C-seq and 4seq was able to outperform all individual tools regarding the median F1 score. And yeah, the results of the simulated data indicated similar trends and results. And this brings me to the conclusion. And yeah, we saw that the results underscore the tremendous potential of ensemble approaches in the 4C-seq analysis. And for the future, this means that the development of more sophisticated ensemble approaches hold a substantial promise for better leveraging the synergies among existing algorithms. That's it from my side, and thank you for your attention.